Hello everyone, my name is Chimeras, I'm here in Compliance, and it's my pleasure to welcome you to our colloquium for today. Our speaker is Orit. She received her PhD in material science in 2012 from UCLA. She then completed her postdoctoral work at Boulder, was recruited to join us here at CU as part of the Biofrontiers faculty research and computational biology. Her research aims to understand the underlying principles that shape collective coordination and communication in complex biological systems, whether that communication is via chemical, sound, or light. Primarily uses insect swarms as a model system for answering questions about these collective behaviors. And her work has produced a stream of eye-popping and fascinating results with implications for autonomous systems, material design, swarm robotics. Her work has appeared in many prestigious venues and has also been covered extensively in the popular press, including by the New York Times and NPR. In 2021, she was selected as both a National Geographic Explorer and she received the Junior Scientific Award from the Complex Systems Society. Please join me in welcoming Dr. Orit.
For this great introduction. Can everybody hear me okay? Is everything set up microphone wise? Okay, great. So I'm super excited to be here today and tell you about what my lab has been up to in the last couple of years. As the title suggests, it's going to be related to communication in insect swarms. But before I dive in, I want to take just a few moments to tell you more broadly what are the kind of questions that we ask in my lab. Hopefully this works. Okay, so we think a lot about animal communication. I think that as humans, it's sometimes easy for us to forget that it's actually really hard to communicate over large distances. We have mobile phones, we have a bunch of backup systems, but animals need to sometimes solve similar challenges with only the resources they have in their local environment. And indeed, many animals evolved lots of beautiful ways to communicate with each other. There's tactile communication, visual, chemical, vocal, sometimes a combination of these modalities. And in my lab, we ask a lot of basic questions about signal design and communication in nature, such as how should organisms choose an optimal signal modality? How should they integrate that information? Sometimes it's temporal, sometimes it's spatiotemporal. And how should they respond? And when we try to answer these questions, we bring in some concepts from physics and computer science, such as detectability, how to tune signal-to-noise ratio, compression, minimization of bits and energy, robustness, how robust the system, the communication system is. And the approach that we use is designing phenomenological theories and models that make testable predictions that then we test with the animals, with real animal experiments. And we sort of iterate between these two approaches until the overall outcome of the model and also the experiments converge. Okay, so we work with a wide range of organisms, with bees, with fireflies, the plants, sunflowers and mimosa. We also work with ferrets and earthworms, and all of these systems straddle different types of communication: visual, chemical, and mechanical. And we specifically work with these systems because, first of all, just by looking at them, by looking at the patterns of their skin, like the bioluminescence, by looking at their body posture, like the bees when they're chemically communicating, we can tell what they are trying to communicate to other members of the group. And they're not just doing this in pairs of two individuals, but they're also doing this in really, really large groups. Most of these systems operate when there's tens or thousands of individuals. And due to the time limitation of this lecture, I'm going to focus on two of my main model systems, fireflies and bees. And what I want to tell you, the outline for today, we're going to start with fireflies. Use visual communication. I'm going to try to answer some questions about how they synchronize with each other and how they decide what to broadcast. I'll also mention some future directions. And then we'll move to talk about bees. There, the communication is a little bit more physical. It's close in range, so it's going to be based on chemical and mechanical communication. And we're going to try to answer questions like, where is the queen? How do they localize the target in space? Why do they create stable aggregations? And likewise, I will also mention some future directions. Okay, so let's dive in to the Fireflies. And just to make sure we're all on the same page, this is an example of a firefly. It has a lantern organ in its abdomen, and it's producing cold light, bioluminescence. And fireflies use that kind of communication, especially during mating season. Each species of fireflies, there's almost 2,000 of them, have a very particular flash pattern. Here I'm just showing you two examples, and as you can see, the males have a very particular on-off pattern of the flash, and then the females also have a species-specific response pattern. So this is, this is an approximation. The flash here is basically either on or off. This signal is very digital in a sense, 0 and 1s. It's really close to computer language in that sense. The problem is that fireflies, when they communicate with these little flashes, they do so. They tend to do so in large groups. If you just do a really quick thought experiment with me, if you imagine one firefly flashing, you don't see when it's not flashing, but you see the next flash and the next flash. It's really easy to connect the dots and then interpret what was the flash pattern that this individual produced. But as soon as you have more than one, and actually this becomes harder and harder as you have more individuals, it becomes a really hard problem to assess which pattern each individual produces. So this is really important because this is how they recognize each other. So one way that fireflies deal with that is by synchronization. They synchronize their flashes, they all flash at the same time. And synchronization is actually a really broad, very common phenomenon in nature. It can happen in inanimate systems as well as animate systems. For example, as humans sometimes synchronize our behavior. And it's really like a textbook example, fireflies are, of synchronization. I still remember using that textbook when I was an undergrad and learning about the problem of synchronization, and that was when I first heard about firefly synchronization. So just to give you a little bit more background about the kind of mathematical models that people use to explain how a really large group of individuals is able to synchronize with each other on a global level. It starts actually with just pairs, and it actually will start with one individual, where the main assumption is that each individual is producing some kind of periodic function. So whether that's a clap, whether that's a flash on or off, it can be described by a phase, with a period. And over here, this is one really simple classical way to couple two of these agents or more of these agents, where theta I is the phase of one individual, this is the period for that individual. It's going to be coupled with the coupling coefficient K to all its nearest neighbors. Now, that coupling K can be lots of different things. It can be a constant, which is the simplest. It can be a function of the distance, which makes sense if you think about fireflies. Their light is so dim, so if they're further away from each other, they're probably not going to see each other very well, and that those going to be less coupled. And it kind of explodes. You know, there's a whole zoo of models. This is just taken from a review, and every time you change something about that whole interaction, of course, it might also affect the global outcome of synchronization. And these mathematical models actually have been really well studied for decades. There's lots of them, but as it turns out, there's not a lot of quantitative data to validate these models for good reasons. Fireflies are a little bit hard to study. Their light is really dim, so until 5-10 years ago, people actually had to go out in the field with photo-amplifying diodes, which is a really big thing, to capture the dim light of fireflies. And also the fireflies only exist as flashing adults for about 2 weeks out of the entire year. So you really have to be at the right place at the right time. But nowadays, just in terms of the technology, we can use really simple off-the-shelf cameras like GoPros, and sometimes even with new smartphones, you can actually capture the dim light of fireflies. And that's what we did. So this was led by Rafael and Julie, who are mentees in my lab. We went together to the Smoky Mountains, where we knew that there's one big swarm of synchronous fireflies, and we recorded them. I just want to play this example. These species of fireflies, Photanus carolines, they flash in bursts. So they flash a couple times on off, and then they stop. You can see that now there's a break, and then another burst is going to appear. And as you, I hope you can see the flashes. Really bright. It's not perfect, and there's some interesting spatial thing going on, right? There's like these waves that kind of start here, go to the bottom. I'll get to that in a couple of slides. But this is really just one camera, and I want to show you the kind of data we can acquire from one camera view. So it's temporal data. And what I'm showing you here is a function that correlates correlations in a time series of the flashes. It's a little bit easier to understand when I plot them. So over here, this is the number of flashes that we get in each frame, and the x-axis is time. We look at a particular time window, and then we look at how many flashes there were. This is what's plotted here on the x-axis, and what is their variance. This is on the y-axis. So we see two different trends. You can kind of see the onset of synchronization, where at small densities of flashes, which is on the left side of the x-axis, we're seeing something that resembles a scaling law that we predict from a Poisson distribution. So there's no synchronization, the firefly flashing in somewhat a random way. But then as the density, the number of flashes increases, we are getting this we can see the emergence of the synchronization, and this is the signature of the burst structure of the entire group. This configuration, of course, will have a different relation between the variance and the number of flashes, and it's reflected here in a different scaling law in that upper plot. So that's one way that we can characterize the onset of synchronization. We show that it's actually a function of how many flashes we have on average in our field of view. Okay, but I just told you that distance can be really important in some of these mathematical models. So to be able to know how far fireflies were when they were flashing, we're going to have to add another camera and do triangulation. So this is two-camera configuration. It's pretty traditional. It's called stereo vision usually. And here we have two cameras that are positioned outside the swarm, and they're looking in. Then once we identify the same firefly with the same impulse views, we can triangulate and estimate how far that firefly was from the camera. We also developed a slightly a different approach where we use 360 field of view cameras, and we put both of them in the middle of the swarm. So in a sense, we're kind of seeing what would be perceived like from the perspective of an individual firefly inside the swarm. This required a little bit of new math, but we developed it, and it's now open access and free for anybody who might want to use it to study fireflies or other things. And we can do this three-dimensional reconstruction. This is one of the first reconstructions that we did. So that was a burst. Now it's the quiet part, and we can start bursting again. And we continue recording this over the duration of the night. So we actually get quite a lot of data points, enough data points that we can start calculating things like spatiotemporal correlation functions that we need for the mathematical models. So this is one example of a spatiotemporal correlation function. The y-axis is the distance between pairs of fireflies that are flashing, and the x-axis is the time between pairs of flashes. So we're seeing a lot of interesting structure here. This is on a more shorter length scale. We also see some spatial correlation on a larger length scale. In particular, I'll first try to describe it in words. So when we're standing in the forest over there with the swarm, what we notice is that we're seeing fireflies that are further away from us starting to flash and burst, and then fireflies that are a bit closer starting to burst, and closer, and they pass us. So there's this interesting correlation and wave of onset of the burst, and we're trying to characterize it here. So over here, this plot is actually all the flashes shown from the top. And we color the flashes according to where they are in the burst. Early flashes are shown in blue, late flashes are shown in red. And in this case, you can see that the blue is a little bit closer to the camera, and it was getting further away from us as we were continuing to record. And one important thing to mention here is that the speed of flight of a firefly is an order of magnitude smaller than the speed of the wave propagation. So that wave propagation has to be explained or accounted for somehow by social interactions that are potentially visual and using the flashes between the fireflies. All right, so everything I just showed you was recording the fireflies outside. These swarms, especially the ones I showed you before, they're really, really large. They span miles over miles over miles. And we're only capturing a small fraction of them in our field of view. So we never really know how many fireflies we have. That can be a little bit hard to account for when we're trying to extend the decision making of individual fireflies. It could be just a firefly just outside the parent queue that we're missing, and it's making an important contribution. So what we did here is actually bring with us to the field these big tents. They're black. So the fireflies that we catch and bring into the tents are actually visually disconnected from the rest of the swarm. We know exactly how many fireflies we have, what their species, what their sex, and so on. And we can also track individuals inside the tent and follow their flash patterns and start asking questions about how they make decisions in order to sequence. So the first questions that we started asking are, we want to really just start with the basics. So we're starting with the main assumption of some of the models that I mentioned before, which is what periodicity of individual fireflies, and can we characterize it? And again, a reminder, so there's two time scales here. There is the time of flashes between bursts, and then there's times between the beginning of one burst and the next burst. So when looking at the short time scale, the time between bursts, between flashes between bursts, we're seeing that the fireflies are doing a fairly good job synchronizing with each other, even when they're in really small groups. So what you're seeing here is a tracer plot. Each firefly is shown in a different color, and you can see that their flashes are somewhat aligned. However, when we look at the longer time scale, so the time between the start of one burst and the next one, something different is happening. So over here, this is the distribution of that time between bursts for a relatively large number of fireflies, 20. And over here, we're seeing that there is a pretty characteristic peak around 10 seconds, which is also what we're seeing outside. But then as we decrease the number of fireflies to 5 and even more so to 1, the distribution becomes wider and wider. And it's got us to kind of question the assumption that maybe these fireflies can be characterized as oscillators, at least on that time scale. So to offer a remedy, Owen, who is a graduate student in my lab, in collaboration with Sri Iruvisas from Purdue University, we tried to find a different model to explain the periodicity, emergent periodicity, without assuming that individual fireflies behave periodically. And it turns out that there's a whole other classical range of models called integrated fire that were originally designed to simulate how neurons synchronize with each other, hence the name, do a pretty good job in explaining or correlating with our data. So I'm just going to really briefly explain the different notations here. On the left side, we have the voltage that's coming from historically the voltage of the neuron. And it's going to be correlated to a few processes on a short length scale, time scale, and a longer time scale. This is all taken from the data that we recorded on the firefly, so we don't need to guess any parameters here. Then we have this element T, epsilon, that tells us whether this individual is charging or discharging. And finally, the only one open parameter we have is beta, which is the social coupling between the individual agents. So when Owen ran these simulations, he noticed that there is an interesting transition where at low beta values that are shown here at the top, we get a pretty wide distribution. This is really just a distribution of that we are plugging in from our experimental data. There's no particular peak or time scale, but then as beta increases, then we're starting to get a characteristic peak at around 10 or 12 seconds, which is similar to what we're seeing in the experimental data. And that behavior becomes a little bit more, the transition becomes a little bit more vague as we reduce the number of individual fireflies, which is also in agreement with our experiments. Okay, so that's all I wanted to say about how fireflies coordinate and synchronize when they're interacting with fireflies from their own species. But as I mentioned, there could be lots of different species occurring at the same place. So what we did next is trying to understand how these patterns, flash patterns of individuals evolved to be distinguishable from each other. And we were really intrigued by these kind of phylogenetic trees. Here you see all the different flash patterns that individual fireflies have and how they evolved. And we were wondering if we could make use of that data to say something about the evolutionary driving forces that perhaps brought the fireflies to where they are today. So this is led by Chantal, who is a postdoc in my lab. Sorry, I forgot to say that this is really similar to Morse code, but you probably already figured that one out. Okay, so what Chantal did is we started with a really simple cost function that we defined for the evolutionary process. It's the simplest one we can think of. It has two elements. One element is defining the similarity between two flash patterns. And here is an example of this. We define it as the longest common sequence that is common for both of these species. Of course, there's lots of different ways to define this, and this is the simplest we can think of. We also did some temporal shifting of these two patterns because there's no guarantee that fireflies will start flashing at the same time. And the second element is we refer to it as energetic cost. So how many beats are on in sense for each one of these patterns can also be thought of as predation risk. So the fireflies, when they're turning their lantern on, they're exposed to predators, and they might want to minimize that. Okay, so to try to simulate the evolution of these patterns, we use a scheme called the naming game. It's a little bit like a Monte Carlo simulation, where we start with a random set of flash sequences, and then we choose one of our species and we mutate that pattern. So we flip one with on or off, and we accept if it reduces the cost of the entire function. And if it doesn't, we accept it in the lower probability. And we just repeat this on and on until the cost function converges. So here's an example of such a simulation. We started with a bunch of sequences. We have 6 fireflies here. And at some point, as you may have noticed, the sequences kind of snapped, and they converge on this pattern. We were really encouraged by these results because we're getting some periodic flashes. We're also getting some bursts, although we did not explicitly put this into the requirements of the system, so it's kind of like an emergent property. Okay, so we can continue playing this theoretical game. We can change things like the ratio between the weights of the two cost function elements. We can change the number of species, and we can see how that affects the cost function. But we can actually bring it back to the data and what the fireflies know are really doing by trying to reverse engineer what were the ecological constraints that occurred while these fireflies were evolving. So in this particular toy example, we're working with 6 fireflies and their phylogenetic trees. The simulation followed that tree, and we generated a bunch of sequences. Then we compared them to the real patterns that the fireflies use, and created this description shift function that tells us as a function of the weights between the different cost elements. So in a sense, we can find the most optimal ratio that gives us an agreement between the simulated patterns and the real fireflies patterns. So you might say that would it be really important to make sure that the fireflies actually coexist, that the species actually happen at the same geographical area, and you will be right to say that. And this is something that we're working on. We're trying to get more data about more species of fireflies in different locations in the US. So we do that in-house, and sometimes when we do that, we actually find unexpected things. One of them is a recent study we had with a species of fireflies that's called pheromone-based. They are flashing continuously throughout the night. And usually it kind of looks like this. It's the same plot that we saw before, the number of flashes as a function of time. They're synchronous, so you can see these big peaks where everybody is flashing at the same time and nothing in between usually. But then at some point in the night, this happens. We colored the flashes that don't follow this particular rhythm in red. So there's this background signal, and we're not sure what it was at the beginning, but it actually has a signature of a chimera state. And for those who are not familiar with that, chimera states are, they arose kind of from a mathematical curiosity of the classical models that I mentioned before. People knew about them since 2004. This is one of the first papers. It was cited thousands of times. And it's really hard to find these chimera states in real life experiments. People are able to sometimes reconfigure special experiments like a system of metronomes and get this kind of configuration where there's a main beat where agents are following the beat, and then there's an off beat that some agents are following that. But this is one of the first occurrences of that phenomenon in nature. Okay. So we're trying to collect more data in-house, but as I mentioned, the fireflies are around for a really short period of time, and we cannot be at the same place. We cannot be at more than one place at the same time. So we're trying to crowdsource the data acquisition, and we have a project where we're using the same method we had with the GoPros. As I mentioned before, it's fairly affordable and easy to use. We just extended it to eliminate camera calibration. And so we're actually using the flashes of the fireflies to realign the cameras. So potential volunteers that would want to help us collect data, the only thing they have to do is go out with 2 GoPros that we send them, of course, and press record and send us the data. And then we follow the analysis that I mentioned before. We can create a database for the species and the behavioral variability, and then train a machine learning classifier, which we're doing now in collaboration with Ryan Lair from the department, to classify which species it was. And we're also collaborating with a bunch of conservation organizations that help us deploy that tool in a more broader level. The last thing I want to mention about future directions for fireflies is that we can also trigger them with artificial light, which is a really convenient way to kind of poke the system, and from the response learn something about the underlying principles. So this is again led by Owen, and what Owen is doing is bringing an LED to the field, exposing the firefly to that LED, and we can train the firefly that is shown here in green to the LED if the frequency of the LED is closer to the natural frequency of the firefly. We can also kind of mess with them and use other frequencies, and over here we're kind of treating the firefly to do something completely different that it not normally do. We can run these kind of parameter sweeps, and the goal is, of course, again, to try to reverse engineer what are the local rules that the fireflies are following. Okay, so this is it about fireflies. I showed you a couple of examples of recent work. We saw the emergent signal periodicity. We also saw the firefly-inspired vocabulary generator, and I told you a little bit about some of the future direction, more species, crowdsourcing, and chimeras. And now I want to move to talk about honeybees. One thing that is important to note about honeybees is that they have a slightly different, or very different hierarchical structure inside these big groups and swarms. So there's only one queen, and there's tens of thousands of other individuals in that colony, but the queen is the only one that is able to lay fertile eggs, so she's really important for the reproductive success of the colony. And the bees kind of tune and try to keep track of where she is most of the time. So the kind of questions that I want to ask now is how the bees find the queen, how they aggregate around her, and a few others. And this is particularly interesting because the bees find the queen using chemical signals called pheromones. These are really small molecules that decay very rapidly in time and space. So bees that are further away from the queen basically have no chance to find her unless they just randomly bump into her. So what they do is generate their own chemical signals, and they use that to inform the other bees. And this is just in terms of posture, how it looks like. This bee has a pheromone gland in its abdomen. It's going to fan its wings to create airflow that drives its pheromones backwards towards the back of its body axis. And this is a project that's led by Dumai, who was until recently a graduate student in my lab. She just graduated. We noticed that bees that sense the pheromones of the queen don't just walk all the way to the queen. They would actually stop at a particular distance and start producing these chemicals. It's called scenting. That's the jargon name for that behavior. And then more bees would sense those chemicals would start scenting, and more and more and more. So they create this really dynamic network of communication. So we wanted to understand the decision-making process a little bit better. Dumai did a bunch of behavioral experiments where the bees were confined in a semi-dimensional arena. We could film them from the top. We also partnered with Greg Stephens to come up with a high throughput machine learning classification code. It was fed a bunch of movies of bees that are scenting and bees that are not scenting. And the output was, first of all, yes, no, is this bee scenting or not, but also in which direction it is scenting, which is really important for interpreting that behavior. So here is an example of an experiment. It's already analyzed, so every time you see the blue arrows, that's scenting event. And you can scroll a little further, a little faster. At the end of the experiment, which is roughly 30 minutes, the bees do find their way to the queen and they aggregate around her. She's right here in this little cage. Okay, so let's stop this. The first thing that we wanted to check was really whether these scenting events are meaningful. Do they really point the bees in the right direction towards the queen? And the way that we tested this was taking the snapshot from the movie, with all the little scenting events that I'm showing here now. We treat them as gradients, and we integrate them into a higher dimensional surface, where higher values on that surface should correspond to where the queen is, if indeed that signal with all these little cues lead to the queen. So this is an average over the entire experiment, and we see that indeed it looks like the bees create this global map that leads them towards the queen. We can also dive a little bit deeper and try to understand how bees make decisions to start scenting. So what I'm showing you here, we follow all the bees that are scenting, and we're finding their nearest neighbors, and we're calculating the distance to their nearest neighbors, which is plotted here, which gives us a pretty specific value of about 6 centimeters. And if we think about this for a second, we're talking about diffusion. We know from the laws of diffusion in physics that a particular distance from a source corresponds to a particular concentration that is coming from that source. And if the bees are positioned at a particular distance, maybe they are really being triggered by a threshold of concentration that gets them to act differently. So to test this hypothesis, we turn into an agent-based model where the bees move in a two-dimensional environment. They move randomly until they bump into a cell that has a concentration that is above a certain threshold. And then they have a decision to make. They either try to climb up the pheromone concentration gradient, or they stop and they scent and they produce that signal by themselves. We have a version where the signal is isotropic. Let's see if I can play this again. And also a version where it's more directed, and it's a parameter in our simulation. So the nice thing about the simulation is that we can test things that we wouldn't be able to do in the experiments, especially these behavioral parameters. So how directed the signal is, which is shown on the x-axis, and what is the concentration threshold that triggers the sensing behavior. We can't really ask the bees to do that, but we can check that in the simulation and see the effect of it. So what I'm showing you here is a phase diagram of all of these different configurations. And we're seeing a couple of different phases in the aggregation process. The first one at high threshold concentration is actually that there's no aggregation, but here's the queen. The rest of the bees are just never being triggered because the concentration threshold is so high. On the opposite side of the phase diagram, we're seeing a situation where the bees kind of get stuck in these smaller clusters. We call this, this is referring to a local equilibrium. So if you wait long enough, they might find their way to the queen, but it's going to take a really long time. Another phase is diffusion limited aggregation. So the bees kind of move randomly until they bump into that cluster around the queen, which of course also scales very badly. You see some size. And then there is this sweet spot where the bees are able to actually propagate information about the location of the queen over the entire system size and also aggregate around her in an efficient way. And if you're familiar with aggregation problems, these phases are not that surprising. But what's really interesting here is that that optimal phase really opens up at non-zero values of the directional bias. So that implies that having that directional bias is actually beneficial for the robustness of that process. All right, that's aggregation in a really clean two-dimensional environment, but of course in reality the bees need to deal with more complex environments. We're testing that in 2 dimensions, either by basically making their lives a little difficult, we put them in these mazes, or just having simpler obstacles. We also just recently ran a classical Y maze where the bees have the option to take the shortest path to the queen or the longest path, and they seem to be doing a pretty good job of actually finding also the shortest path in these unconstructed environments. And this is in 2D. We actually study bees in 3D. This is a saga that started actually a long time ago during my postdoc, studying these kind of structures. There's nothing but bees in a tree branch here, so the bees are really suspended and they need to stay mechanically stable. We showed previously that when we apply mechanical perturbation, which is a nice way to say that we were shaking the bees do change their structure. We can relate that to mechanical signals that the bees might be sensing inside the swarm. But everything we knew about these swarms was coming from looking at the swarms from the outside. So you can see the shape of the swarm. We can see maybe bees that are kind of moving on the surface of the swarm. But if we do a really quick back to the envelope calculation, most of the bees are inside, and it's, gets more and more important as the swarm increases, and they could have a big effect on how the swarm is being stabilized. So more recently, Olga, who is a postdoc in my lab, figured out how to work an X-ray machine, and we created the first ever construction of a honeybee swarm inside an X-ray machine. So we have access to the entire distribution of bees inside the swarm. And we can start asking questions about how the bees are arranged inside to maintain mechanical stability. So we started analyzing this data, and we thought about an analogous problem that is actually relevant to individual animals, in particular humans. There's a really well-known scaling law that relates the strength of an individual to its body weight. And this is actually taken from a paper from the 50s, where people use records of weightlifting competitions. It's like really well documented apparently, and they know how to draw these scaling laws. And we tried to look at something similar, but for superorganism. So we did something a little bit different. We took a slice throughout the swarm, and we measured how many bees or what's the weight of that slice. and how much weight underneath it, which is this part over here, the rest of the swarm, how much that weight this slice of things is supporting. And we're seeing that these plots actually do follow the universal pair, so there is also a scaling law here. And scaling laws are great because now we can predict the distribution of these over several decades. And it's important to note that all these different colors are actually swarms of very different sizes, and they all follow this universal curve. So in collaboration with Kaushik, who's here in the audience, we started looking at the bulk structure, trying to interpret it, and I don't have time to get into all the math, but I will just say that we created a force balance equation for each layer and how much weight it means supporting, and we can actually solve that equation. This is the resulting equation. We can solve it numerically as a function of that exponent that we have in the scaling law. So what I'm showing you here is the solution. That's the black line. How much beads are in each layer as a function of where they are in the swarm. This is for the exponent that we know is the exponent from the experiments, and it's in good alignment with the experimental data that is shown here in gray. And we can also play with this change numerically the value of the exponent and see very interesting shapes of the swarm. For example, this blue one is a swarm that has very little mass at the top and very large mass at the bottom. Of course, that's not physical, but actually we have some preliminary results where we see that these compromise that optimality to potentially deal with other stressors like shaking them or changing the temperature. Okay, so the last future direction I want to tell you about is a little bit of leap. So we saw how beads aggregate. We saw how that happens in 2D and how they maintain mechanical stability in 3D. What we're starting to do now is actually trying to understand how bees, while they're in the swarms, manipulate inanimate matter. And this is referred to as collective construction. How we all know that bees create these beautiful honeycombs. They're highly regular, but it's not really well understood how the bees do that. How do they each deposit a little bit of material to create these really large scale structures? So this is a project that is led by Boulder, who is a student in my lab, and partner with Francisco local Chimeras, who is from the aerospace department. And we're again doing the usual thing. We're trying to annoy the bees. We're trying to create a configuration that is not going to be able to solve that problem perfectly. In particular, where 3D printer starter frames kind of look like these. So there are two lattices, but they're just a little bit off. They're not perfectly aligned, either in terms of angle, stability, or distance. And we're seeing that in the experiments that Boulder is running, the bees create some defects at the interface between these different lattices. And this is actually really interesting because we can reproduce the same defects using a really simple model of aggregation of inanimate material like carbon layers. And this work is going to come out, I guess, in a few weeks in the NIS. Okay, so to summarize what I showed you with bees, we saw how they use chemical and mechanical communication. We talked about the signal directionality in the chemical communication, how it's important for the aggregation process. We also talked about the mechanical stability of three-dimensional swarms, and I talked a little bit about future directions like collective constructions and perturbing these swarms. And I hope that together with what I showed you just before about fireflies, that I was able to convince you that these are really interesting systems to look at. They are all operating in a distributed way. They had eons of evolution to perfect some of these algorithms. And just as an outlook, I want to point out that I think that by understanding how nature solves these kind of problems, it might also have implications for technology, for swarm robotics. We have a few of those here in our department. And by better understanding the biological systems, perhaps we can design technologies to rely on these evolved tools. Okay, that's it. The most important slide. I want to thank my awesome lab members, consisting of postdocs, graduate students, beekeepers, undergrads, and a few really, really talented high school students, all my collaborators and funding, and that's it. Thank you so much for your attention.
Right, let me just start off. I was very interested in when you're talking about stability of the mechanical stability of your piece, but let's talk about the stability of these oscillators, right? So eventually they synchronize with each other. I wonder if you study like the stability of those kinds of synchronizing oscillators in fireflies, like do you actually find any evidence that this will always be stable or?
For more mathematical models, that usually depends on the parameter space where you are. You have stable solution and non-stable solutions. For the actual data, we're seeing that there's a lot of variation all the time. For example, in terms of the wave that was propagating, that changes. It's very dynamic. It changes in time. It also changes in direction. I can just pull this out for a second. The Chimera states that are also known mathematically to be sometimes stable and sometimes not. Where is that? They're not really stable over the course of the night. So what you're seeing here on the y-axis is the fraction of synchronous fireflies to the ones that are offbeat as a function of the flash density. And this is early night, so it's very disorganized, very non-synchronous in a sense. And then as their density increases, they actually they increase a little bit in their synchronization, but it's not perfect. They only reach better synchronization once their density decreases. So this is a very I think it really the answer is depends, and we have lots of evidence of non-stable states.
Yeah, great talk. Thank you so much. I have a question about the citizen science component that you've done here. Can you talk you talked about as crowdsourcing, but it sounds like it's not, because you actually have to interact with them quite a bit to make sure you get the data you want and give them the equipment. Can you tell us a little bit about how you infrastructured that, how big it is, or if it's how ongoing it is, how standalone it is? That'd be interesting to know.
I think I might have here. So right now, this is it.
Okay.
These are our volunteers that helped us this year. And they're actually come with mixed training. Some of them are actually entomologists. This guy is just a photographer who knows where the fireflies are. So we are in a stage where we're kind of beta testing the method.
Sure.
We already improved it a lot just based on their feedback. And we're in contact with Professor Sarah Lewis from Tufts University. She's like the world expert on fireflies behavior and conservation. And she just over the last couple of years started some initiatives for firefly conservations for actually, as it turns out, once you look at it, surprise, surprise, they're also actually suffering, and they were red listed by the conservation organization. So we're trying to collaborate now using these methods on a broader scale. We have some plans to continue that in the next experimental season. So it's not ready to be deployed like globally, but that's the hope.
Did you find the people first, or did you need certain locations and then you found the people who were residents in those locations? Because fireflies, we don't have. Do we have them in Colorado? I don't think so.
Oh, we do, yeah. So
Okay.
there's a myth that they don't exist here.
Okay.
So that's the thing with fireflies. I think there's probably lots of places where they exist and we just don't know of,
Okay.
because they need darkness.
Yeah.
And they're also around for a very short period of time.
Right.
So kind of related to your question you do need somebody that knows they want to be there and has some statistics. So that's why crowdsourcing is potentially a good idea, because Maybe there's lots of places where fireflies exist and some people know they do exist there, but we just don't know because we don't have that data. Yeah. So,
Thank you.
yes.
This is going to be an embarrassingly unsophisticated question on your opening slide and the last one of your examples of a swarm with sunflowers. What's the nature of communication among sunflowers?
Okay, so totally unrelated, and I'm sad that I don't have any data slides here, but I can tell you a little bit. So the sunflowers, in the specific case that we work with, communicate using visual signals. So plants in general know if they're shaded by another plant because the light has to go through the leaf, and that changes the spectrum that comes out, and plants know how to recognize that currently. Versus a more fixed configuration, like if it's a cloud or if it's a roof, and they respond differently when they're shaded by other plants and by more inanimate objects. And in particular, we're looking at really dense planting configurations of these plants. And if you plant them in one line, they start growing, and then at some point they kind of snap with those zigzags. One goes to the right, one goes to the left. To us, this yells immediately like Isaac model, you know, that. So there's mathematical models that are really intuitive and relate to them very deeply. And so we're collaborating to experiment with and also doing computational modeling to try to understand how they make decisions of where to move right or left and avoid shading.
Thank you. So lifelong gardener, that's fascinating.
Yes, yeah.
In the experiments that we're doing with the LEDs to manipulate the fireflies, which sounds just like really cool. Like how much control do you like? Can you make one firefly species respond using a different species signals?
Yeah, that's kind of the hope. So then we can test the sensitivity of that individual species or that individual firefly within the species to other patterns that we know exist. For example, it actually, it's just a side note, there's a lot of mimicry in nature in fireflies. So there's actually species that use deceiving signals to talk about fireflies. And in particular, it's females of a particular species that mimic the flash patterns of females of another species. They lure their males potentially for mating, but they just eat them. And we're really interested actually in testing some of these sensitivity and interactions between patterns or from different species in that configuration. See there and then copy.
Okay, so yeah, I might have missed this, but is there a relationship between different environmental models like temperature or season, and like how are they related with the fireflies synchronization?
Yeah, so first of all, both of these temperature and humidity effects, they have a big effect on when the fireflies are going to emerge. In terms of synchronization, it turns out that temperature can also change the frequency of individual fireflies. They flash faster when it's warm. So you can kind of imagine that we haven't tested it, but that would be one reason to do that, because they will change some parameters of their individual behavior.
It was really cool to talk with. So I wanted to follow up on the Chimeras signals you're talking about. And I was curious if you have hypotheses as to the ecological significance of some of these. Do you think these are like anti-predatory that's really cool. I mean, but something like a LED or robotic device
exactly.
might be able to test.
Exactly. Sure.
So maybe one more question. So you create a lot of these mathematical models. I wonder, like, how many of them are actually amenable to any kind of analysis, or are we just going to do numerical simulations to learn more about the properties
analysis in terms of
Like
comparing-
just trying to solve the, I mean, I know the differential equation would be hard to solve, right? But just trying to prove something about the models, is there hope that we could say something about those models through purely mathematical methods, or do we just have to run lots of simulations to find out what they're doing?
Are you asking whether it's feasible to do purely analytical theory versus
Yeah.
numerical or agent-based models?
Yeah.
My intuition is that analytical theories work for really simple situations, and it's much easier to explore with agent-based models. So we actually start with agent-based models. That's usually the case. And then if possible, we're trying to build an analytical theory that connects to the agent-based models. But I find it's not intuitive to me to come up with an analytical theory just upfront and connect that to the data.
Let's thank Orit again. And please feel free to take some cookies or drinks on your way out.